Hey guys, it's AIM. So this begins the start of the Chapter 7 lecture videos, which are optional. You don't have to watch them, but if you need a little help with the material, anyway, they're here. So in Chapter 7, we're basically talking about how do we murder microbes, okay? And specifically the pathogenic ones. We'll also get into this in Chapter 20, using drugs inside patients, but this is more vocabulary driven, and it talks about general microbial control. Okay, so like I said, there is a bunch of vocabulary. Okay, so let's go through some of these terms. Okay, so sterilization is one when we want to destroy everything. So whether it's a bacteria, a virus, fungi, endospores. So this is basically when you use a method like an autoclave, okay, which is high pressured steam, and at a very high temperature where you're trying to annihilate everything. So obviously this is not something that, you don't put a human being inside an autoclave. This is when you really want to make sure that you have gotten rid of everything. The interesting thing, and I mentioned this, is an autoclave can't get rid of prions. And we're going to talk about prions in Chapter 13, but they're just little hunks of protein. They should be denatured, so I don't know why the autoclave can't get rid of them. But basically, sterilization is defined as destroying all microbial life with the exception of prions. Okay, disinfection, and I'm sure you're used to this term, but disinfection is what you do in your bathroom and your kitchen and when you are putting your Vesphine on your lab table. So this is when we are using a chemical to try to destroy most things that are there on an inanimate surface like a countertop or in a patient room like patient bed rails and surfaces that you would wipe down with a disinfectant. Okay, so it's using a chemical, but it's not as intense as using sterilization. You're just trying to get rid of most microbial life. Antisepsis is doing the same thing as disinfection, but you're doing it on living tissue. So think of doing like an alcohol wipe before you give an injection. Okay, that would be an example of antisepsis. And you have to use chemicals that are less powerful because you're dealing with living tissue as opposed to an inanimate surface. Decontamination is more of a general term. So this includes not only using a chemical, whether it be a disinfectant or like hand sanitizer, but when we say mechanical remover, that means that you're also scrubbing. Okay, so you're using a chemical in combined with scrubbing or some type of physical process. Maybe it's mopping a patient bed or a patient's room, the floor of the room. So that is an example of what decontamination is. So when we talk about the resistance to death, basically, of microbial control, we want to get rid of the following categories, okay? So bacterial vegetative cells, that means bacteria that are just living their daily lives. And remember, some species of bacteria produce endospores. So those you're not going to get rid of with disinfection. If you want to get rid of endospores or cysts from protozoans like Giardia, that's when you have to use an autoclave, okay? You're not going to get rid of those with just spraying a disinfectant on it. But you can get rid of other types of fungi, meaning hyphae and spores. Hyphae just means fungi that's growing. Yeasts, which are unicellular fungi. You can destroy worms, you can destroy viruses. And remember, those nasty little hunks of protein, which we call prions, are resistant to all of this. So they're resistant to disinfection, antisepsis, and sterilization. So sterilization, as I mentioned, is when you want to get rid of everything. So bacteria, viruses, cysts, endospores, everything. And mostly performed with heat and pressure. That's what the autoclave does. Sometimes when it's something like the inside of a building, which, is obviously too big to fit in an autoclave, then you can use really severe chemicals that are used, and those are called sterilants, and they're basically just pumped through whatever building you want to try to sterilize. But the key is that if you're going to sterilize something and be able to call it sterile, like think of surgical tools, instruments used in the operating room, they have to be able to stand up to heat and pressure. And so this is always reserved for inanimate objects, and it's specifically reserved for inanimate objects that can handle high heat and high pressure. Okay, but that's the goal, is to remove basically everything except for prions. Okay, disinfection is when you're going to use a chemical called a disinfectant to try to get rid of most microbial life, and definitely to destroy vegetative pathogens, meaning that these are the things that are actively living, like living bacteria, any viruses that may be on that surface, any living fungi, like yeast cells. So disinfection too is less severe than sterilization, but we only disinfect on inanimate objects and surfaces. So it will also destroy any type of toxins that might be secreted from the cells. So a typical disinfectant might be, say, a 5% solution of bleach, which is something you don't want to rub on human skin, right? That would be really caustic and it could burn the person. So disinfectants are much more powerful than antiseptics. Okay, and antiseptics are trying to do the same thing as disinfection, but you have to use chemicals that are less caustic because you are using it on living tissue like human skin. So think of betadine, which they rub on a person, scrub a person before they have surgery. Or there's another chemical called chlorhexidine, which does the same thing. So you're trying to create, as sterile an environment as you possibly can, but you have to be careful because you're dealing with living tissue. Okay, I also want to mention the next two terms because you'll notice if you put an A in front of the word sepsis, it means something completely different. So when we say a person has sepsis, that means that they have microorganisms that are in the blood and tissues and tissue fluids, and those microorganisms, usually bacteria, are actively multiplying. Okay, it used to be thought that sepsis was always an end-of-life condition. It's not. A person can be septic and come back from it when they're treated with antibiotics or antifungal drugs, but it's a pretty serious condition. Okay, so that's what sepsis is. And then if you put an A in front of it, it completely changes the meaning. So asepsis is good. It's something that please promise me that you will always follow asepsis during your careers. And it's basically any practice that prevents spreading infectious microorganisms into sterile tissues, whether it's your own or a patient's, or bringing it home to your family. So asepsis includes any practices like wearing PPE, following hand washing guidelines, wearing a mask, using hand sanitizer, any infection control practices that you're asked to do at your future career. That is asepsis. So yeah, those two words mean totally different things. Okay. The other thing that I want you to understand is this is a suffix, okay? So whenever you see the word side at the end of a word, it means to kill. Think of homicide, right? Okay, the killing of a human being. So a chemical that is a bactericide is there to kill bacteria. A fungicide kills fungi. A viricide, because viruses aren't technically alive, we can't say that we're killing viruses, but we're going to inactivate them. Okay, a sporicide will kill or destroy endospores. And then kind of general terms that are used are germicide and microbicide. So those are meaning agents that, chemicals that will kill microorganisms in general. Okay, and then when you see the suffix static, what that means is to basically, you're not killing everything, but you are preventing any further growth. Okay, so a bacteriostatic chemical will hold the bacteria at bay and prevent it from growing any further, but it doesn't kill it. Okay, something that is fungistatic will inhibit or prevent further growth of fungi. And so chemicals that say they're microbistatic or germistatic, those are chemicals that are used to not kill everything, okay, but they will hold it at bay. And I will explain why you would use a chemical that is cidal versus static, okay? Most drugs that are taken inside your body are given to you in static concentrations. And we'll talk about why that is in chapter 20. Okay, so I'm going to stop on this slide. Here's our first chunk of the lecture because I want to keep these in about 10-minute segments so they don't get overwhelming. Okay, so we'll pick up with this slide next. Thank you.